And we're back with the final part of the Pickle Lab. Now, let's just recap. We've taken our pickle, we've pinned him to the dissection tray, we have done our observations of the pickle, we've determined which is the anterior, the posterior, the lateral, the dorsal, and the ventral, and we have cut him on the ventral side. We've determined the ventral side is the lighter side simply because it was never exposed to the sun. We did our eye incision where we opened up the two sides of the incision like a door. We've determined that the seeds face from the side in towards the middle, all the way around the barrel of the body of our pickle patient here. We have taken out samples of our seeds to examine under our camera microscope for the class. We've taken out a sample of the skin, the interior tissue, and we have taken a seed out that's covered in tissue so that we can take a little bit better look at it. So that brings us up to speed on where we're at. Okay, the next page here says, use your probe to carefully remove the seeds without removing the flesh. Record your observations. So we notice the seeds face into the center. Seeds are translucent. In other words, we can see through them because they've been in the vinegar concentration for so long. There is liquid present within our pickle. There's an outer skin, there's inner tissue, and the pickle was 15 centimeters long, 4 centimeters across the diameter. Questions. Labeling the following parts of the pickle: anterior, posterior, dorsal, ventral. We have done that. Does the pickle have bilateral or radial symmetry? Well, bilateral means if we cut it in half. Radial has to do with it being around. And it has both, depending if you cut it when you've got it laid out, where you can cut it straight down and get the lateral symmetry. If you cut it where it's curved, of course it's not going to be quite laterally symmetrical. Radially symmetrical? Yes, if we look at the way the seeds are laid out inside, they go all the way around. So if we cut it across the middle and we just like bite it in half, yes, it is radially symmetrical. Listing safety precautions to take. Well, of course, we're always careful with our sharp instruments, the way that we hold them and use them. I have gloves on, and I also have my safety glasses on, even though you can't see my pretty face on the camera there. Then we have the last questions. Now, I've left this blank because if you're using this Pickle Lab, I'm not going to do the entire Pickle Lab for you. But it says, why is it important to record your sketches and data carefully? Well, that goes back to the function of the scientific method. You should be able to answer that. If the pickle were an animal, if our pickle patient were a pickle animal, what would be the function of the exterior surface? This one, my sixth graders actually had some issues with and had to be kind of prompted a little bit. But what is the exterior of you do? And then finally, number 6 here in complete sentences. Write what you learned in this lab. I always like to have my students write at least a paragraph of what did you learn here. Did you learn how to use the tools? Did you learn to observe? Did you see things that you hadn't seen before? Was there something else that you learned in this lab? Did you like the lab? Did you not like the lab? I always want my students to be honest with me when we're doing our laboratories in class. And this concludes the third part of our 3-part series on the pickle dissection. Now, of course, I'm going to turn the camera off and I'm going to clean up, put all of my tools away, make sure that they're sterilized before I put them away, and then we'll proceed to the next laboratory. And I hope you enjoyed it and learned something.